Hello?
Hi, can you hear me?
Yeah, I can hear you.
Oh, great, okay. Yeah, I haven't, I got assigned a bunch of things by my professor, and he's being very picky, so I didn't get a chance to do anything.
Oh, okay.
Uh, and there was a paper I wanted to send you, I think. Well, I sent you one that I found.
Okay.
Is it on worm bits? No. You cut up a Planarian, and each piece is moves independently.
Yeah, okay.
Have you seen that one? No.
No, I haven't.
Oh, okay.
Did you send
Well,
this one?
let me see if I can see it. I don't know which computer I put it on to. Yeah, I don't know. Oh, there. Planarian fragment there. I couldn't find the paper.
Okay.
And so I just copied the reference. Planarian fragments behave as whole animals.
Oh, wow. Okay. Yeah, I've heard of
Yeah.
this where, like, you have, well, the general paradigm where you have, like, you can cut it, planaria up into different parts and they all sort of are new planaria after a while. They regenerate the entire organism. Their cells are what they call totipotent.
Yeah,
Yeah.
the fragments behave as whole animals. Anyway, would you like the reference or?
Yeah, actually, can you share your screen or to show the paper? Or, oh, you can't.
Okay, well, maybe.
Okay.
Find out how to share screen.
Should be at the bottom, the third over from the left, third button.
Okay. My internet, this was being slow again.
Okay.
I'm going to be really happy when I get fiber optic. No, it just doesn't want to.
Oh. Do you have like a link to
Like
the
it.
citation on the web or something like an NCBI stub or something?
Here, I'll just let me. I'll just put it in the chat.
Okay.
Okay, I don't think I copied the journal. But anyways, yeah, I don't know.
There we go. All right. Yeah, that's Planarian fragments behave as whole animals. Yeah,
and then it's got some of the authors,
okay.
various bits attached to authors, like 2 and 1. Anyways.
Well, that's good. Yeah, Planarians have what they call totipotent cells, which mean that you can take a single cell and it'll regenerate an entire organism. And some of the questions they've asked about this is like, well, do they behave like their ancestral animal or their source animal? There we go.
Okay.
And okay, it's not going to work like that.
And then they say data and code availability. So apparently, if you look it up, there should be something.
Yeah, let me share my screen.
Let's see how this one is.
Yeah, so I got it.
Yeah, my screen doesn't appear to be an option for me.
Okay. Yeah, so this is the paper. I'm not familiar with any of these authors, but this is published in Current Biology. Highlights touch and near UV stimuli elicit 3 regionally different Planarian responses. When cut into pieces, mid-body and posterior fragments respond as intact animals. Turning behavior overrides elongation and contraction. So this is, one of the questions that they've had is, like, since the cells are totipotent and they generate new animals, do they behave like their ancestral animals? And there have been some studies that have been done where they've shown that there's some sort of memory that's carried over between the cells. In other words, if you train a Planarian on some task, like some simple behavioral task, and this is what they're referring to here, that Planaria can learn things like simple associative learning. But then you can take that Planaria and break it up into its constituent cells and put those cells down in media, and then you can get the you can get whole organisms to regenerate from those, and they'll remember what they learned when they were like a singular Planaria. So it's really interesting, and this looks like some of the stuff they're doing with looking at the different types of behavior. So this is actually interesting. What is this graphical abstract? So this is the turn, elongate, and contract. So they have the different parts of the body here. And what is this? They don't really have a legend for it, but this is the idea here. So if we go to the abstract here, in recent decades, behavior has been used as a readout to study Planarian development and regeneration, wound healing, molecular evolution, neurotoxicology, and learning and memory. The Planarian nervous system is among the simplest of bilaterally symmetric animals, with an anterior brain attached to two ventral nerve cords. And so this is here, and interconnected by multiple commissures, which is a division, or these are connections between them. So there are two parts here, and there are selected connections called commissures. Where are we here? We found that in response to mechanical and near UV stimulation, so this is with like simple touch or simple light stimuli in the UV band or near UV band, head stimulation precedes turning, tail stimulation precedes contraction, and trunk stimulation produces mid-body elongation. So there are three different types of behaviors when you stimulate them in different parts of the worm. So you would have like you stimulate it here, it's elongation, here it's turn, and then this relates to this network here that you see in this graph, where you have a circuit of turn, elongation, and contraction. And then there's some turn down here, some turn nodes down here. But so this is
I found that.
So then they're able to do this in this certain species of Planarian. When cut into 2 or 3 pieces, the anterior end of each headless piece, so they're cutting off the head, and they have 2 or 3 pieces. So I guess they're cutting it at the turn, elongation, and contact boundaries shown down here. So when they do that, the anterior end of each headless piece switched its behavior from to turning instead of elongation. So this part here, the head of this red part turned, like it changed its behavior to the turn behavior. So it responded as if it thought it was a head. In addition, posterior ends of the head and mid-body pieces sometimes produce contraction instead of elongation. So in other words, these posterior ends here would behave as if it's like the tail. So basically, if I cut this part out, this red part, it'll start to behave like the entire worm, with this end turning and this end contracting and the middle elongating. And then as it matures, it will take those, it'll be assigned those different behaviors accordingly. So that's interesting. It's not just that the cells are totipotent. It seems like the entire organism is has this sort of architecture for knowing what the shape is. And actually, Mike Levin, who we talk about a lot in this group, he's done a lot of stuff with Planaria, and he actually he's done a lot of experiments with bioelectricity and how there's a bioelectric signal that's global in these worms, and that it seems to sort of guide, like if you have a single cell and it regenerates an entire worm, that there's some sort of like body plan there that it can like draw upon to, assemble from a single cell. So this is maybe a similar thing where there's some bioelectric control that sort of sends signals and guides some of these, which end is up, basically. Because if you just take a little piece of it and you throw it off, how does it know which end is forward and which end is reverse? And I'm sure that there are other sort of chemical signaling mechanisms as well. I don't think people really know exactly what's going on, but this is a fun read. The observations show that each mid-body region reads the global state of the organism and adapts its response to incoming signals for the remaining tissue. Selective lateral incisions show that the changes in behavior are not due to non-selective pain responses, that the ventral nerve cords and cross-connectives are responsible for coordinating local behaviors. So there's this nervous system that is in the worm. When you cut it, it reconfigures itself, but there's enough of an architecture here so that it can sort of change according to what it needs when it's cut. And then our findings highlight a fast functional reorganization of the Planarian nervous system that complements the slower repairs provided by regeneration. This reorganization provides needed behavioral responses for survival as regeneration proceeds. So that's that paper. This is the graphical abstract. So, this is a nice paper. Thank you for pointing that out, Susan.
Okay. I'll turn off my...
Oh, okay.
Well, I'll turn things off so that there's more bandwidth.
Okay, all right.
Okay.
Hello, Jesse.
Morning.
Morning. All right. We're just talking about the great world of planarians, or flatworms. And we found a paper on, or Susan found a paper on regeneration. And I don't know if you've read some of the stuff on that, but that's pretty fascinating. Especially, I know Mike Levin, if you know some of his work on planarians, has done a lot of work on regenerating planarians and how they can sort of figure out what the morphology should be from a single cell regeneration situation where you have like a single cell and it can regenerate the entire worm. So, thank you for attending. And for those of you on YouTube, we'll be putting these readings in the Slack, just as a reminder, the OpenWorm Slack in the DevoWorm channel. Okay, so I wanted to talk a little bit about some of the things going on this month. We have our Hacktoberfest is ongoing. We're not getting a lot of participation, but we are getting Ujwal, Tharoon, and Asmit were featured at last week's meeting, and they were talking about some of the stuff they're doing with Basilaria. So they're doing some things to further this digital Basilaria project that we have going on. They're working on a new data set of images, and they're working on a new algorithm called Pix2Pix, which is an interesting algorithm where you map from your image set to some sort of mask set, and you use that as sort of a training model, and then you can use that to find a lot of different types of, like, what they're trying to do is they're trying to create a skeleton of rectangles out of the cells, and then they want to use that as a way to identify cells over time. So they want to they take these movies of motion in the microscopy images of motion. So you have this series of images where, the cells are moving back and forth in the frame. And they want to find the cells more effectively in that type of setting, where you have to identify them, maybe if they're cut off by half, if they're off the frame, or if they're, kind of moving in a weird position, if they're in a weird position as they move, how do you find those? And so they're working on that. Hopefully they'll be able to show some results in a few weeks. But And for those of you who want to participate, I'm going to share my screen. So if those of you who want to participate in Hacktoberfest, I think this is it here. Okay, so this is the digital Basilaria repository, and we have the Hacktoberfest badge. So if you want to, I don't know if there's a lot people can participate in here. Ujwal and Tharun and Asmit are working on this project, and maybe afterwards we can use some of the data to do some interesting things. So you might want to inquire. I don't know. Well, we have some issues here that might need to be addressed, but they're kind of older, so I don't know if they're going to be of use. Then we also have DevoLearn. So DevoLearn is of course, the platform we've been working on for the last 2 years. And we have the DevoLearn software, which has another release coming. I don't think it's been released yet. I have to get back in touch with Mayok and Mainak on that. We need to touch base. But other than that, I think this is something that if you, you can look at the issues and see what's there. I'm not really sure what there is right now that's outstanding that can be achieved in a short period of time. We're working on a paper for this, but this is something that I think it's going to be amongst, maybe 3 or 4 of us to do that. And so, but in DevoLearn in the organization, we have other things going on. We have the data science demos, which are always a good, we've had probably about 8 people contribute data science demos here. And if you have a topic that you want to cover as a notebook or as some sort of tutorial that you can put on the GitHub and have the notes available for people to follow along, then that's something maybe you can contribute. So that's DevoLearn. And then I wanted to kind of cover, okay, so I think that's it for this. That's Digital Basilaria. We have data science demos. And then we also have the group meetings repository, and that's not as straightforward because I don't think we have a lot of issues, but those are mainly on our project board. So you can contribute by going to the group meetings. You can go to the project board here, which is this group meetings projects 1, and you can look over some of the many issues that we have open here. So we have probably about 60 issues open because there are a lot of things unfinished. There's things that are off the radar, there are things in progress, action items, things that we're holding. So things that are on hold, some of these are just kind of things that we're talking to other people with, things that people were already engaged with, but they don't have time to finish now. So, and then to do. So there are a lot of things we can follow up on. This virtual developmental worlds is interesting. There's also, the replaying the tape of life and trajectories. There are a lot of themes that we've covered this year in this set of meetings that I'd like to maybe pick up or move forward. We kind of have a couple weeks to do something. What was that?
Sorry. What was the virtual developmental worlds?
Oh, this one is, I think it's not loading for me. This is the one where we're looking to build a virtual environment or a 3D set of 3D environments for developmental processes. Maybe we were talking at one time about building, 3D models of embryos or putting something into the Blender container for OpenWorm. But I think this is a more general theme where, we're trying to imagine sort of 3-dimensional worlds for developmental processes. So that could be 3D modeling in Blender, or it could be some sort of VR experience, or it could be, just a 3-dimensional model or a higher dimensional model of development. There are a lot of...
Is it related to the Stanford paper I'd mentioned yesterday, or is it more not so much agent interactive environment, more just 3D spaces to represent specific structures?
It could be. Well, yeah, the thing you showed me there is an area they call morphological computation, which it might be useful to do something like that. Where we had if we had like little embryos that were evolving in a environmental context. And we've talked about this, where what is environment? So you have a lot of different things that environment contains. So it could be like a fluid environment, or like a sort of gelatinous environment, or it could be like other types of environments where there's stimuli coming in. So it's that might be an interesting thing to do, like have almost like a fitness based thing where you have like an embryo in a world, and then it has like a physical environment where it's not really exploring the environment so much, but it's exposed to an environment with like light, with surface properties, with heat. Then there also where we could have like little agents that are very like sort of hatching out of their egg, and they have like no experience in the world, and they have to sort of find their way out of an egg. I'm just thinking here about like how if an organism, sometimes organisms are pretty well developed coming out of their egg, but sometimes they're not. And so, what are they encountering as they first enter the world? Those might be interesting questions too. I don't want to restrict people's thinking. I was thinking, maybe more of like a simulation, but I don't think that I want to limit it to that, because I think there's a lot there to explore.
Oh, sure. I don't want to I'm not trying to mesh everything together. I was just, for my own understanding, trying to differentiate them.
Yeah. So yeah, there's a lot of interesting.
Oh, also, just as you mentioned genetic algorithms or something.
Yeah.
I don't know if they reached out to you yet, but this is more for the other group. Ankit Grover DM'd me yesterday about stuff.
Okay.
And he asked me about one of the things, and I said to mention, I said to follow up with you. I don't know if he did. I think he's going to try to do it in Slack.
Okay.
So just a note that he was going to talk about like one of the switching papers or something. I think so.
Okay. Yeah, that's great. Yeah. Probably Slack is best.
Yeah.
I'm having trouble with the OpenWorm email right now, so I don't know if people are trying to send me mail there. Don't do that right now. Just send it to the maybe my Outlook account, or Slack is good too. So I think that's good. I wanted to mention a couple other things here. Let's see. So we talked about this. So it looks like NASA's really kind of going in on open source, and it's nice to see, because NASA's a large government organization, and that like usually they're the last ones to do something like open source. But this is their new open source science initiative, and they're not like funding agencies are a different issue, because they want to encourage the community to do things. But NASA's kind of a self-contained thing, so like they don't have any real. They kind of do have incentive to do this, but because there are a lot of private agencies now that are doing space exploration. So this is their open source science initiative. So NASA is making a long-term commitment to building an inclusive open science community over the next decade. This includes the open sharing of software, data, and knowledge, algorithms, papers, documents, ancillary information, as early as possible in the scientific process. The principles of open source science are to make publicly funded scientific research transparent, inclusive, accessible, and reproducible. Advances in technology like collaborative tools and cloud computing are enabling this, but it also requires a culture shift to a more inclusive, transparent, and collaborative scientific process, which will increase the pace and quality of the progress that we see. And so they're developing this open source science initiative, and then this is where they, kind of list the things that, so it's transparency, the results should be visible and accessible and understandable. Inclusive science processes and participants should welcome participation and collaboration. Open accessible science, data tools, software, documentation, publication should all have this fair standard, which we've talked about in a number of meetings, which is findable, accessible, interoperable, and reproducible. And so that's good that they have those standards, because that seems where a lot of people are moving towards, those specific standards. And then open reproducible science, which is scientific process and results should be open such that they are reproducible by members of the community. This is maybe the most important one. So if you're like trying to do something, not only just like space exploration, but NASA does a lot of exobiology work. And so they're interested in sort of some of the, maybe they do a set of experiments on some extremophiles. Those protocols should be open and accessible. And so they're starting this off this month. And so they're kind of making the case for open science, and they have some resources here for the different directorates here, including citizen science and high-end computing. So a lot of it is earth science and planetary science, but there's other stuff going on there as well. I'll put that in the chat here. And then, so that's that.
I wonder if they were like the diatoms in space that Dick did a while ago. He proposed to put diatoms in space to see how it affected their growth, because their growth depends on their microtubules.
Right.
So I wonder if he'd like to revive that or hand them that.
Yeah, I don't know if they have I don't know if that includes how you're putting experiments on the space station or something, but that would be interesting.
Yeah, well, yeah. That's what it is.
Yeah. I mean, maybe they'll start putting those, if they do some experiments in space, they'll be opening up those data. So they've done this with a lot of different organisms, where they've because people have the similar questions to that, what happens to cells when they're exposed to zero gravity, and what's the growth pattern. So they've done this in plants, I know, and they've done this in other organisms. So yeah.
Yeah. Well, this was quite a clever idea, because if you disrupt the microtubules, they don't develop the holes or the pores in their frustules. It becomes more of a clumped process. So, anyways, he put this together. He applied to the Canadian NASA branch, and then they said, "Oh, well, we're not doing biological experiments from Canada." It's just like,
Yeah.
Yeah, I went to a lot of bother for nothing.
Yeah.
Anyways.
Well, anyway, yeah, so maybe we'll follow up. Someone can follow up on that if they're interested. Let me go to the next order of business, which is submissions. So this is our submissions document. And again, there isn't a lot on here new. I think the Boring Billion and Kindle books are kind of on hold. Mathematics at DevoWorm is also kind of on hold, although thinking about that recently, but that's something we'll kind of talk about in coming weeks. We have these molecular level simulations of diatoms and movement simulations. That's something that I think our group that's doing the digital Basilaria might be able to address with some of the data they're collecting. So once they, perfect their algorithm and they kind of go through some of the data that they have, we'll have some numeric data that we can then look at and analyze. So we might actually be able to address these fairly soon. This quantitative comparison of archaea and shaped droplets, I haven't heard about that in a while. I'll have to check back on that. I think my NACO's working on that, but and then this neuromatch submission. So I think most of the activity on that's going to be from the other group that I work with. You can only have one submission per person, so we're kind of full up on that in terms of ideas. And but I would still recommend that people attend NeuroMatch. I was also thinking of putting on like a parallel session with either DevoWorm or OpenWorm kind of topics over, during NeuroMatch. So it wouldn't necessarily be sponsored by NeuroMatch, but it would be concurrent with NeuroMatch. And I don't know, maybe we can, get the word out that this is happening. I don't know exactly what it would look like. I've been thinking about it for a while. Like, what would this look like? Would this be something that we would have like a call for, and then people would say, oh yeah, I like development and I like the brain, and let's do something around that. And I don't know if it should be like, I don't think it would be like a hackathon, but it could be like something like that, where people just come and try out ideas or talk about ideas. Or it could be something like just a presentation of some of the things that we've been doing. I'm not really sure how to do that. I don't really want it to be like an advertisement for like, it's kind of hard to, people get disappointed in that if it looks like an advertisement. So I don't exactly know. But I would like to do something. And I've been meaning to do this like every NeuroMatch. I said, well, we should do something with, with advertising DevoWorm or OpenWorm. And aside from having like a presentation on it in one of the sessions, it's hard to really kind of get the right mix of whatever it is that you want to get and get the word out. But yeah. So yeah, that's what we're working on in terms of submissions. Kind of like the same as it's been in stasis. I haven't seen too many opportunities come forth for other types of things. Again, the nurips, if you're interested in nurips, that is coming up in the I think the conference starts in early December. So I think this was these were the submissions for the papers and the abstract. So that passed a long time ago. The actual event itself, I think, is in early December. I don't know what the date is. So we'll just, whenever that comes up, we'll be able to fill that in.
Your IPS is December 6th through 14th.
6th to the 14th, okay.
And as far as NeuroMatch goes, I think that would be a pretty good way to do some things. I haven't really thought about what it is, but I feel like there's like you could probably do somewhat of a joint. Yeah, I don't know how much. I guess in the other group we're doing some specific stuff with it, so that'd be more what we're doing there. Yeah. As far as like DevoWorm goes, I feel like it would be in an ideal sense, it might be a sort of like a way to discuss or like a relevant discussion of topics, but also things that could kind of dovetail into actual projects we want to do or would be doing in like going into the next year, like what's going to be happening in January, February, March. It was kind of like the pre Google summer code period.
Oh yeah,
so that was something to think about.
Yeah, like I usually do. Well, I try to do a yearly recap at the end of the year, but that's pretty much close to the end of the year anyways. So yeah, going into I guess that could be. And then the question is like, how do you get people to notice it, or is it something that would be like concurrent with NeuroMatch, or is something maybe we pre-record and put up during, I don't know the logistics of that. What would be best?
Also, actually just thinking about some open science stuff, like I know, and this is something that came up in a DM mostly in the other group, but like it may not necessarily be a specific, like, it may not be biology related, but whether it's this group or the group or OpenWorm or whatever, but like having an open science like discussion or like part of that open science project, like we were going to do videos or something, having an open science itself is like the NASA thing we just saw, or reminded me of it. Having an open science like I don't know, seminar or whatever might be, or, something along those lines may also be a good time, because NeuroMatch is very on board with like open and accessible stuff.
Yeah, I think that, yeah, and then that might be a good idea too. Have an open science component. Just kind of like going over some of maybe the way we do it here, and then, maybe a more general way of doing it, like getting people familiar maybe with the INCF resources and some of the other resources that are like more neuro-oriented. Some of the other things that, like with OpenWorm. So OpenWorm, I mentioned this, I proposed this during the OpenWorm week annual meeting, and they were like, oh yeah, it's okay. I don't know how much we're going to get participation out of the different groups. I don't think it's going to be like this. I think it's something you have to organize really far in advance if you want to get people's attention. But we could definitely do here's OpenWorm, and I did a talk at AWIFE 2020 on demystifying OpenWorm. Because there's been a lot of press about the singularity and all this. That apparently there's also an OpenWorm Ethereum cryptocurrency, and I don't know. I don't think it has any connection to the project, but it's out there, just so people know. So yeah, it's taking on its life of its own, but people won't know the core of what's going on. And I know that actually I mentioned that Stephen Larson gave a talk last week. So I watched, this was after our meeting last week. I watched it and it was pretty good. It went over a lot of the sort of the history of OpenWorm and what, sort of how it's progressed, some of the things that have been accomplished, looking at simulating ion channels, simulating worm movement, in physical environments. So people have done that at a very high level. And there are a lot of participating labs in conjunction with OpenWorm. So a lot of those are in Europe. And, a lot of the C. elegans labs are always interested in this. So it's I don't know, maybe I think it's underutilized, the component where you say, okay, I want to get involved in OpenWorm. It might actually be relevant to my work. Because a lot of people think, well, it's this thing where it's like they're trying to simulate them, singularity or something, and people, maybe don't think it's relevant to what they're doing. But it's actually maybe highly relevant to a lot of people's work, especially if they're doing C. elegans, but sometimes even if they're not. So, I think that would be good to have that kind of, maybe like a demystifying session.
For open science in general, or OpenWorm again?
Oh, probably for both, yeah.
Okay. I had one more tangential idea that just came up, or more so a question. And it's kind of interesting because it's about the context of my question is obviously I'm applying to graduate school and updating my CV, and I'm rearranging a bunch of subcategories in the CV, and the sort of nebulous questions about service and how you show off different things that you're doing. And I'm in the process of doing that, and this came up a lot last year when I was doing a bunch of virtual conferences during the summer. And I remember, I might have this more in the other group, but I remember saying things about oh yeah, it's really important. People are starting to want to see your contributions to open science and to open source code and things like that. And I think I have a question of how maybe it would be in a service, under a service heading in a CV, but how and where would you list your contributions to open science or things like that? Because I haven't really seen that specifically, but I also feel like I'm hearing, I've just thought of it now, I haven't really looked into it,
Yeah.
but I also feel like in some of the feedback that I've gotten, and also just to kind of say it here, I'm for those who don't know me about it, I'm part of a fellowship program, and part of the fellowship program, is doing these mentoring sessions and discussion sessions with the cohort of the people in the fellowship. And one of the things that came up in one of the sessions was, like, it's great to show specifically your diversity and inclusivity activities in applications and in your CV or however you're addressing that for graduate school, because that's a very important thing. And there was talk about that and some things about do that. And I thought, well, we didn't talk about yet, and I'll bring up next time there, but is the open science component. Like how do you address that or label that even? So I'm just thinking about that out loud right now.
Yeah, I think there are a couple of strategies for that. One is that service is usually like, working on committees or working on like a, project where it's administrative. So yeah, I think open science falls in a couple of these different traditional categories. You might want to have an open science category you can do this on your CV, just put open science activities, and then you put those in. And I know a lot of the things you've done, like with Neuromatch and other things with the orthogonal lab are kind of non-traditional. Like, people generally don't do them. It's not something that everyone gets exposure to. So that would be good under open science activities, because it's not really service. Yeah, but it's not really anything else. It's just kind of its own category. So
I'm almost, for me personally, because I for a long time misunderstood the service category, and then I talked about it with the mentor, and it was sort of like like what I did for hosting the CogSci panel, like that kind of stuff, or being a moderator, the CogSci discussion group, and then a bunch of the, like, I've done panels like with New York Celebration of Computing. I've done my alumni stuff. I've been a moderator for something with you, and elsewhere I've been moderators. Like, those kind of all, okay, that makes sense, and like even specifying what those are, and like getting used to categories and subcategories. But then I'm thinking like, I'm just now considering like an open science thing, and also potentially it might just be open science and or like kind of a mentoring, or not. I don't really want to say professional development, but that's sort of adjacent to what we've been saying in the other group. So there's sort of like, it's a mix between those are things that I'm doing, and also I have a lot of stuff that I'm trying to cut down, because I don't want my stuff to be too long. Even though CVs are supposed to be long, like, I don't want to bury the lead about anything. So I'll show you more later. This is kind of just things I'm thinking about in the moment right now.
yeah.
But I appreciate this discussion. So.
Yeah, no problem. Yeah, I think service is like very broad, so you can just have subcategories of it, and people know kind of where to put it. Just as long as it's like, something that's a coherent heading and that you're building on a theme. So, I've done a lot of mentoring. I've done a lot of like open science service or to that community, things like that. And then, you can have whatever categories you want. Really, I mean, I don't know. I'll probably share some of my CVs. I have like a main CV, which is really long, and then I have some alternate CVs, which are shorter, which focus, like, I have one that's open science, one that's like just selected publications. So there are a lot of different like ways you can tweak it, just for the, like, because you'll find that like when you write a grant, for example, you have to have a very short report of what you're doing. Like, it has to be like 2 or 3 pages. So but then also you want to have a longer one so you can show people what you've done. So having like this, maybe different versions where it's modular, you can take things and cut them out or put them in depending on what you're applying for, is good to have.
Yeah, I know, I think Morgan actually mentioned this last time. I'm sorry about the background noise right now. I'm transitioning here. But like, there's like, do you ever hear about the OpenCV project? Or like the OpenCV, or AutoCV, excuse me.
AutoCV, yeah.
Like, did you ever use that?
No, I'll look into it. I just heard about it like Saturday, so I'll look into it, but I've not heard of it.
I'm not really sure what to make of it. Basically it's an attempt at combining your Orchid stuff and like automatically pulling from that sort of, and some other adjacent things, which makes sense. But also, like, I wonder if I kind of haven't really invested that much into Orchid, but I wonder if, for funding purposes and everything else at this point, it might be better to put things on there because that's like a very, this is tied to like your NSF stuff, right? Like it's very, so yeah.
Yeah, that's just usually for, well, you can put publications. Yeah, you can build stuff on there. Yeah, I would do that. Put everything in. There was a period where I was putting them into all these different tools like Orchid and all these. So sometimes you have to be redundant and put things in different places. I definitely update my Orchid profile though, because I'll use that for like NSF and NIH related stuff.
I think that's a lot of like grant stuff and like even Neuromatch, I think, and other higher level stuff seems to use that as like a credible source of like, we're going to look at your profile and have you connected to stuff and everything.
So do you need to update your Orchid, like, or doesn't it automatically update what you've published, or? It
usually automatically updates, well, the publications if they show up on like NCBI, but if there's specific things, you can actually update it like a CV, like different like presentations and service things too. So you can, just to make sure it's updated though, because it usually auto-populate, but you'll have to update it manually to make sure.
I've been ignoring it.
Well, I mean, some people like it. I mean, I'm just saying that it's useful in some cases. So.
Yeah, I'm basically putting my full CV on Lakes to be super professional now, and I was looking at Auto CV, and Morgan mentioned it, and I feel like I'm on the fence about it, and is it how much is the juice worth the squeeze kind of a deal for it, because it's sort of setting up your own thing, but also I have to put, I don't have anything in Orchid right now, and like some things I need to have. It's a nice conversation, and we don't have to talk about it anymore here. But like, just a lot of, it's a very interesting time for me. I feel like between now and the end of the year, I'm in this sort of, zone of expedience and very careful choices about what I try to do.
Yeah.
With my time. So yeah.
Okay, well, yeah, if we have any questions about that, we can follow up later.
Yeah, thank you for the discussion. I appreciate it.
Yeah. So actually, I wanted to share some more, a couple more things here, and then we'll move to papers. This is I want to go over this because this is maybe tied into, what we might be doing in the future. This is something actually that so at Networks 2021, and then I did another presentation on this at another workshop. There's this idea of embryo and developmental networks and how they're sort of divergent from one another in development. So you have like a bunch of cells in an embryo, and then you start to get neurons that differentiate. And then those neurons will wire into a neural network, and then the embryo cells will differentiate into different tissues. And the question is that when they're in this embryo of maybe 200 cells or 1,000 cells where there's no differentiation except maybe the germline, they're all interacting with one another, signaling, and then all of a sudden you get these differentiated cells, which is, their own thing, and then they start to sort of behave sort of as a group of cells. So, when you get maybe some sort of critical mass of neurons, like maybe 10, they start to wire into this network. And when they start to wire into the network, they have their own behaviors. They interact with one another. They affect one another through signaling. The same thing when a tissue starts to form. That tissue is sort of become self-contained and modular. And the question is can we characterize this formation of modules in the embryo with networks or network theory? And so I've been working on this for a while with different, presentations and publications that are available on this. Euler Cycles for Life Talk is also kind of included in this because it kind of deals with this issue of emerging modularity. We have some papers that been published on the emergent connectome and the embryo networks work. And so there's some open data sets. And then what I wanted to bring people's attention to is that, I'm kind of building this document. And I know that Jesse and I have talked about these kind of documents where, it's not really a review. It's not really a survey. It's sort of like laying out this theoretical approach. And to do that, you need to have like a summary of past work. You need to have some basic concepts. You need to have a review of like what kinds of things we're talking about. So we have conventional types of complex network, random, scale-free, small world. These are all kind of like the complex network types. We have this experimental control network, which is described in the literature specifically for brain networks. And then you have different types of spatial networks. So spatial networks turn out to be important here. We have different types of spatial networks that we can consider node and edge construction. So this is how you take a bunch of nodes, which are cells, and how do you connect them? Using what criterion, but more importantly, using what kind of set of rules. So, you can use something called preferential attachment, where you attach to the most highly connected cells. So, this sort of reinforces the idea of hubs in a network. Equiprobable edges is where you build a random network just based on, any edge being equally probable. So it's a random process, or a fitness model where you look at different connections and consider their fitness. So these are all important issues. Topological minimization. So for brain networks, the idea is that circuit needs to be minimized in terms of its connectivity. And if you go into embryo networks, that's probably important too. So you have a number of different ways to do that. And then, there's a listing of some of the main issues. So criticality in the embryo. We have a couple of different ways of looking at criticality, wearing minimization, some references there, network structure, some standards. So this is an INCF standard on standard representations of network structures. And then a bibliography, which is kind of what we have here, but this is a more general bibliography. I bring this to people's attention because, this is a kind of a document that if you're working on an idea that you can't seem to write, or before you start writing a paper on it, and you have an idea, you have a project that you want to work out, this is a good way to do this. You start with like some publication or some presentations. Maybe you have a publication on something related, and you plug those in. Then you have these open data sets if they exist. You have some concepts, you have some key ideas, concepts that feed into what you want to do. And then, maybe I haven't added in here, well, this is kind of like experimental stuff, but there may be some experimental models that are appropriate. And then, maybe some concepts from complex systems or computer science that you might want to bring in. And then, some references where you have like very specific topics that you want to review, and then some standards that you can draw from. So this sets up everything. You have, now what I want to do with this is I want to write a paper more directly on this topic. And so I've been kind of collecting this for 2 years, and I'm interested in writing a paper on, or a preprint on this. And I think it's a lot clearer now than it was a year ago. But having this resource here, I think, can help people guide their way through this area, this topic. And I know so this isn't really a review. It's kind of not really a proposal. It's kind of like a way to guide people through this topic, which is actually quite difficult to understand. And we can build on this. So these are stubs that, you can just say, why is wiring minimization important? Why is network structure important? Maybe, we can consider these different types of criticality in context, which ones make more sense, which ones make less sense to describe changes. So when we have like a neural network and an embryo network, when they split, when they diverge, is there some sort of criticality that's driving this? Is there like a neural avalanche or a jamming, which is like where you have a bunch of particles that kind of get to a certain density and then change their phase behavior? There's something called propagation criticality, and then symmetry breaking. All these things may have more or less to do with this phenomena. We would like to know, we would like to evaluate those. But to do those, you need to identify what they are. And so we have all those things here. And so I'm just pointing this out so that people are aware of this kind of document. And I think this is useful for any idea if you want to work it out. So that's the link to the document. And also, if you're interested in this topic, we can talk more about it. I think maybe this is something that could be of interest to the NeuroMatch crowd as it deals with kind of like emergent neural networks and things like that. So the next thing I want to get to is this. Okay, so I'm going to talk a little bit about regulation and gene regulatory networks. And yeah.
Before you move on, would this have anything to do with consciousness? Your GitHub?
I don't know about consciousness. I mean, I think that's maybe something to do with it, but I don't think it's the main idea. I mean, like maybe eventually you could talk about it that way. But
okay.
Yeah.
I know someone who has a PhD thesis on this, and he might be interested. That's what I'm asking.
Oh yeah, definitely. Like brain networks and that are there's a sort of an issue there with like the emergence of consciousness in brain networks, and there might be a link there. So
okay, well then I'll give it to him then.
Yeah. So there's this. I wanted to get into this topic. So this is a book, or we've talked about gene regulatory networks before, like the way people have built them as computational models, and we've talked about them in biology in the context of like genes being regulated. But there's actually this book that is Eric Davidson wrote back in, I can't remember how old this is. This is not a new book, but it's, The Regulatory Genome, Gene Regulatory Networks in Development and Evolution. Eric Davidson, who's now deceased. This is a scan, so it's kind of sideways here. I don't remember.
Okay, I can't see the screen.
Let me share my screen. I'm sorry. So this is Eric Davidson. This is the Regulatory Genome, Gene Regulatory Networks in Development and Evolution. So you can see this big circuit here. This is a gene regulatory network in the cell here, and this is just one set of genes that interact, and this circuit describes like some process that's being regulated for the purposes of producing this phenotype or some phenotype. You have a lot of arrows, a lot of lines, a lot of like arrows that go up like this. So this is from 2006. So his work, he had been working on this, I think, for like 30 years before writing this book. And some of the things I'd like to point out here is that you have these different chapters in the book. So chapter 2 is inside the cis-regulatory module, control logic and how regulatory environment is transduced into spatial patterns of gene expression. So a cis-regulatory element is where you have the gene and then you have the regulatory elements right next to it. So it's within a single unit, hence cis. And they talk about these different principles. So it's very closely tied to spatial patterns of gene expression. So you have these circuits that sort of describe, this promoter is driving the expression as gene. And then, you have this gene product. Sometimes it gets regulated by an enhancer, so it gets tuned, its output. But all these things are very specific in space because eventually what's happening is a phenotype is being built. And so, maybe cells in a certain part of the embryo are being, have this one expression pattern, and in another part of the embryo they have a different expression pattern. Could also be that, within the cell itself, there's spatial, so maybe something is expressed out at the membrane, but not in the, so much in the, localized in the cytoplasm, these sorts of things. And so there's this downstream of specification, spatial repression, and then the power of the cis regulatory module. So this is where you have diverse regulatory outputs from a simple input. So this is where you have a simple input, but because of the way that that regulatory element is structured, you can have different types of outputs. And so this chapter talks about the operating principles for cis regulatory systems. And they talk about some of these in pluripotent embryonic cells. So specification processes by which these cells choose among diverse fates are allocated to give spatial territory or enlage. To understand the role of cis regulatory elements and specifications necessary to think of them as devices that make choices. They produce alternate outputs depending on the sets of inputs which they are confronted with in different cells. So this is, there's a figure down here. Yes, this is it. So this is cis regulatory control of even skip, the even skip gene in Drosophila. And the even skip gene is where you have this pattern, this spatially restricted pattern where you have stripes. And you have, depending on where the cell is in this pattern, you have this network where you can have, so you have this set of genes that are sort of aligned on the chromosome, and they're regulated in different ways. Some are repressors, some are, some are expressed highly, so these stripes here are the dark ones, and the repressors are the gray ones, and you can see that the activity is enhanced in these stripes and repressed in these darker areas. And these are, I think we talked about Hox genes before, these are collinear on the genome. So these are lined up on the genome, and they're, expressed depending on their location in the cell's location in the embryo. And so this is another example here where you have spatial repression of the regulation of this gene here. So this is where you have different target sites and spatial inputs. So you have spatial inputs to the model, and you have different target genes, and they get expressed depending on their spatial location. And so this is something that is a probabilistic process. So this doesn't happen in every embryo in the same way, but it's, there's a stochastic component to it, but it behaves in a way that does produce patterns. And so there are a lot of ways you can design these. These are in nature, so they're not designed, they are actually evolved. And you can see how these things kind of co-evolved over time. So there's this type of, so this is a model of cis regulatory expression in Drosophila. The short range repression, A through D. So A through D is where you have short range repression over space. You get these spatial patterns. And then E through F are long range repression, where you get a long range repression over space. And so I'm not really doing this, I'm not really getting very deeply into this, but I wanted to show you some examples of what these look like. So this is, this is one chapter, and then the other chapter here is this chapter on regulatory hardwiring. So this is a brief overview of the genomic control apparatus and its causal role in development and evolution. So there's this regulatory apparatus encoded in the DNA, gene regulatory functions and development, genomic regulatory sequences, and the evolution of morphological features. So there are a lot of these. This goes back to sort of this problem of diversity of form in animal life. So by the end of the 50s, it was clear that the causal differences between body plans of a fish and a fly, or a sea urchin and a mouse, are somehow encoded in their genomes. But this fact alone does not provide the mechanisms by which various animal forms arise in development or arose in evolution. So a large part of this answer lies in the gene control circuitry encoded in the DNA. So this is something that, again, this is a graph of egg RNA complexity and genome size. And you can see that, like in this case, genome size, as it increases, it sort of has this relationship with RNA complexity, although in the area of 10 to the 6 genome size, in terms of kilobases, there's a lot of variation here. So there's some relationship between RNA complexity in the ag and genome size, although that's not entirely. It just shows that, there's a lot of, how much transcript is produced versus how big is the genome. And so the idea is that there's a lot of, the more RNA you have in the egg, the more regulation that's going on. So that's what they're trying to show there. You also have gene numbers and genome size, so you see that in general there are fewer genes than genome size. There's a lot of genome that is, outside of coding regions of the genes. So you have a gene and then you have maybe junk DNA, or maybe you have regulatory elements. And in some species, that's a lot of stuff, and in some species, it's less so. So there may be some relationship between, if you have a lot more genome in terms of genes or base pairs than coding regions, that at least a portion of that is regulatory. And so that's kind of an interesting set of relationships. These are different species. I think they just, so CE is C. elegans, for example, where you have this, you have genes and then the size of the genome. And that is actually maybe similar to Ciona intestinalis, or the C squirt, which is also something that has a similar mode of development, but actually has a much more complex adult phenotype. So you can see that, like, the relationship between the adult phenotype and the the number of regulatory elements that might exist in the genome are, there isn't any strict correlation there. So, overview of the regulatory architecture. So this is another example of a regulatory architecture, a modular cis-regulatory information processing module. So this is where they show kind of the space-time domain 1, space-time domain 2, and then these other inputs. So you can see that you have these different modules that are regulated by different signals. So you have different spatial repressors, different signals from adjacent cells, cell cycle control, which is something controlled by maybe some other genes involved in cell cycle, which is a cyclical process, different lineages, lineage identities, which we've talked about in our meetings with respect to C. elegans. And you have this, essentially it's a mapping between these two modules. So you have one module that has one set of inputs, another module has a different set of inputs, or they're mirror inputs. But the point is that these two modules will then feed in to this final module here, and this is an OR function. So this can be like a logic gate, basically. That each of these modules can be, you can have one module operating or another module operating, or both at the same time. And there's an OR function here, which means that it could be either or module feeding into this. And in this case, they're all wired in the same way, so they're basically redundant. And this is actually important in evolution because, to have multiple modules that can do the same thing. It can step in if one module is, say, removed by evolution, or there's a duplication of something, and the circuit is sort of modified in some way, that these redundancies can be very useful for maintaining the right amount of RNA output here. So, this is also the case in where you have developmental mutations or ablations, where things are, maybe deliberately shut off by a experimenter, or maybe they're shut off by some, developmental disorder or mutation. And so that, in that case, the redundancy that you have here is good. But the idea is that there's this complexity of different signals, repressors, and other things that feed into this and then produce the RNA output. And that RNA output then will produce proteins and a phenotype. So there are a lot of regulatory demands of development. You're executing, they take the view that it's the execution of a genetic program, that the program is the concern, and the program is maybe looks something like this. And so this is the thing we want to be concerned about, and understanding that wiring blueprint. And so pattern formation, we talk about pattern formation a bit, and thinking about pattern formation maybe in this way versus like a purely physical model is really interesting, because you're basically making, arguments that either physics is the main driver or genetic regulation is the main driver, but of course you can have physical influences on that, and it can regulate this. So, this is kind of like almost like a cybernetic model in a lot of ways. And so if you think about it like that, it's kind of an interesting take on it. And so these models that I'm showing you have not been implemented in biology or in computation. They're biological first models, which means they've taken them from the biology and mapped them out using experimental methods. But people have really struggled to try to make these computationally salient and scaling them up. So, so this is cis-regulatory elements of some muscle-specific genes. You can see that there are a very diverse range of elements that can be used to regulate the expression of different genes for different functions. So in this case, for muscle. And so you can see across species that you have these modules that are, kind of aligned on the chromosome that enable a gene to be regulated in different ways. So you have all these different regulators that either can suppress things or they can tune things, like an enhancer, or they can just, act as like a binary switch. They can either turn genes on or turn genes off. All those things exist in these. This is a very diverse type of model for sort of controlling gene expression and controlling the production of a phenotype. And so I think that's all I wanted to say about that. There's also a review article that I found on this. This is Eric Davidson from 2005 and colleagues. This is computational representation of developmental gene regulatory networks. So as in Eric Davidson's later years, he tried to implement this in silico in computational terms. And they actually did work on this biotapestry, which supports the process of model construction and also model visualization. So we have some, and we've seen this in the A-Life community too, where people have tried to make these GRNs salient. And a lot of them are very primitive. They either look like this, which is a hugely complex circuit, or they're very primitive in terms of what they can do. But there's some principles you can use to build on. So for one, you have developmental robustness, which means that you have multiple mechanisms for the same thing. You have multiple positive and negative feedback loops. So there's robustness. You also have other things like redundancies. So this is where you have multiple things that do the same thing, but it's like if you knock one out, it seems like it's redundant at the time. Maybe you have like 10 genes producing the same gene product. And, that may seem like a waste because a lot of the product actually goes to waste. But if you were to have a situation where 8 of those genes got knocked out, then suddenly it makes more sense. And this is the thing about evolution. Evolution is this open process. It's this open-ended process where there's no real goal or there's no real design objective. You just have, things just have to, they kind of get generated, and then if they're successful, they're more fit, and then they persist over time. And that's exactly what you see with robustness and redundancy, is that robustness is in terms of function, redundancy is in terms of sort of structure, and they give you these options as the organism moves through time. So they kind of go through this. They say that we refer to the GRN state in a given cell at a given time as the view from the nucleus in that cell. Each view from the nucleus describes the set of genetic regulatory interactions specific to a particular group of identical cells at a particular moment in time. So this is an interesting way to think of this. There's also the view from the genome, which is a summary of all interactions in all cells of interest over the entire period of interest. And so VFG does not display time or space, but only has one copy of every gene in any of the VFNs, and summarizes the genetic potentiality of the embryo. So there's this potentiality that we have in the embryo to produce a phenotype. And in this case, they're using these different viewpoints to characterize them. So you can either have a view from the nucleus, which is where you have these different interactions. It's sort of more of a cellular view. And then the view from the genome, which is more of a genomic view, where it's like looking at the cells instead of the genes. And so you put those two together and you get this. I think we talked about like cell regulatory networks a couple weeks ago. We had a paper on that. And then we also had a paper a long time ago about GRNs in silico, where they build models of these genes. So this is their platform where you're building these complex structures, and this is for like, chicken embryos or mammalian embryos or other types of embryos that aren't C. elegans. And you can see that they can build these models in the software and, map out these interactions. I think the step forward, though, is to build something like a GAL4 UAS system, a computational version of this. And if we can do that, then this is like going to be the breakthrough in this area. This is the GAL4 system. This is a very famous experimental system. This was developed in the 80s and 90s, actually, were refined in the 80s and 90s, where they basically used, this is in fruit flies, where they used this GAL4 element, where you can you separate out like different parts of the gene expression process. So you have, the system has two parts, the GAL4 gene encoding the yeast transcription activator protein GAL4, and the UAS, or upstream activation sequence, an enhancer to which GAL4 specifically binds to activate gene transcription. The benefit of a system like this would be, in biology they use fluorescent proteins, but the benefit of this in a computational sense is it partitions these two processes. It partitions the process of activation and then also the process of what happens upstream in terms of activation. So it tells you kind of like what's happening at the enhancer and at the other regulatory elements. And then it also tells you what happens upstream, what gets activated. And so if we divide it into those two components in a computational sense, we can understand this process better. We can build better models of this for different things. And I know we talk about genetic algorithms, but this is sort of a variation on this, where you have these regulatory sequences, where you have genes that maybe evolve through mutation or recombination, and then you have these modules that regulate them and tell you how much of a certain gene product you have at a certain time. And then that can lead to building like maybe simple phenotypes. But to have this type of system in place, you want to be able to like unpack what's going on computationally and experimentally. And this is one way to do this. So I wanted to leave that there. I think that's a good sort of pointer to the future. I don't want to say that this is something that has been solved. This is something that needs to be solved. I think it has a lot of utility. But yeah, I think that's good. And so I'll leave it there. Susan had to leave, so thank you for going, or thank you for attending, or Jesse had to leave. Okay, sorry.
Okay, well, I'm here. I really like these papers. That they're interesting.
Yeah, it's good. Okay, well, thanks for attending. Talk to everyone next week. And if you have anything you want to talk about, email me or Slack me or whatever.
I'm still trying to work on active matter.
Okay, yeah.
Yeah,
Hopefully we get an update on that soon. Yeah.
Yeah, I'll keep trying.
All right.